Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we will learn about how to use PyMOL in Google Colab session for high quality images. Before this, I have covered several videos like how to work with PyMOL in offline setups and how to install PyMOL into your local computer. But I felt that most of you guys are unable to install or to work with PyMol. So therefore, I brought this tutorial for you that how you will be able to make good high quality images for your thesis, research, presentation, and other things without installing PyMol. Okay, let's quickly cover this slide. What are the purposes of this tutorial? After completing this tutorial, you will be able to generate publication quality. From the publication quality, I mean extremely high quality images. So to generate publication quality images for your thesis, for your research papers, for presentations, for assignments, reports, and web pages, if you are managing or if you have a web page or in HTML, etc., etc. So you will be able to generate high quality images using PyMol in Google Colab setup for free. So let's start. First of all, you have to use this link. I will drop this link in the description section of this video. It will lead you to my repository. Once this link is open, you will see this kind of window. Okay, here you need to select PyMol. Right now it is the last one. You will see this kind of file here, generate high quality image with PDB to PNG PyMol. I just forgot to mention that the specific package we will use in PyMol in a line session that is PDB to PNG. We will convert the PDB file to PNG because the PDB is the file which is open, which can be open in PyMol. It's normally a protein files or protein DNA, whatever, but the format is PDB and the picture format we will generate is PNG. So click here. Once you click here, then you need to click here, the connect button. Okay, the first part of this tutorial is just like my introduction, the introduction of this tutorial. And if you have already PyMol installed, then I would suggest to click here. And these are two high quality tutorials and it will help you a lot. But if you are unfamiliar or if you are not familiar with PyMol or do you have some problems in the installation, although I have also covered a video for installation, you can see on the channel. But if you still have some problem, then you can use this tutorial for some of your work. Okay, so now here you will need to provide your protein. I have just selected this protein 4EXS, but you can change it to your protein from the PDB. Let's say you want to 4I5S. Okay, you want to work with this protein. Let's say I have changed it. This is another protein, histidine kinase of the two component system. Then you need to click here and click here run anyway. Okay, now here we will do some installation stuff and remember this installation stuff will take some time, maybe from 4 to 5 minutes or maybe a little longer, depending on your internet setup. So let's install, and here install PyMol without Conda. So we are installing it without Conda environment. So therefore it will takes a bit longer, 5 to 6 minutes probably. So for the reason I will just pause this session. Once the installation is complete, I will come back to you. Okay, welcome back. I have finished the installation and it took almost 7 minutes. Okay, you can clear this. Now we will import the required libraries into this workspace. Here, we will also import the PDB to PNG already installed in first cell, but please do not clear this from here because this section or these information will help you a lot to generate figures. So now here we will use a PDB. So in the start we have used a 4i5s. So here you need to provide your PDB name again, or if you want to change it, let's say, okay, here we use 4i5s. Now edit this name here, 4i5s. Please try take care, do not disturb the structure of this script. So click here. The This script will download, or I would say import this structure from the PDB. So it may take almost 1 or 30 seconds or 1 minute. But once again, it depends on the internet connection you are using. Okay, so we will generate figures after this step, and I will also teach you how you can tweak different things during the generation of different styles of figures. Okay, it's finished now. Everything is ready. Now I'm going to generate a simple figure first. Click here. Now you can see. So this is the figure we generated from the first code or running this first cell. Okay, now just click here to save it. Let okay. After every figure you generated, the next step I kept will work for saving that figure in high quality. So just wait. Sorry. Now you can feel how high quality it is because it's taking a bit longer in downloading or in rendering the picture. Generation is not a problem for PyMol, but exact generation normally takes some time. Okay, now how to download this figure? Click here. You will see PDB to PNG. Click here, and here is your PDB which is downloaded, and here is your PNG. So just click here, let's say download. Okay, these three dots. Now the PNG is here. We will check whether it's fine or not. Oh, okay. I got this one. I will tell you why I got this one and uh, how you will get the exact one. Okay. So this is, we call it black and white. So this is one of your figure. Now, how to change the question is, okay, I got this because I use BW. BW stands for default and black and white. Okay, I will remove this and will save again. I will teach you how you can tweak or how you can change such kind of things. First, let me show you that yes, we can save these kind of images in our workspace or quickly for our presentation and tutorials. Okay, what I have done, I have removed the BW option here. So we will wait. Okay, done. Now click here, refresh because this picture will be changed to a new one. Yes, here it is. So you can see it's sufficiently high quality. Okay. Now the last question, it can come in your mind that how we will change to different styles, to different orientations, to different colors, to different showing ligands, each and everything. So how we can do that? Okay. So how you can tweak all these things? Okay. If you have cleared this thing already, so you can run this cell again. Now, just pay attention here. This is the main thing which gives you the information for different things. If you want to rotate proteins, then you will provide some information, let's say 250 comma 50 comma 50. So what are these things? These are actually the degrees. I means here you can see 250 comma 50 comma 50. It means on x-axis the rotation should be 250 degree, on y 50 and on z 50. So once again it will be some trial and error method because fixing rotations on x-axis and y and z-axis may be a bit hit and trial method if you are interested in the rotation. For the color, we mention names of the color. Let's say here it is red. Okay. If you want to show ligand ID, then what you will do? So we will just say ligand chain and in which chain it is located that way. So here is the complete um, how to say script informations, what you want to show, what if you want to increase the height, decrease the height, if you want to change the render style. Let's say you want to know how many render styles are present. So come here, render style. In render style we have default. Default is the one we have generated now. We have default black and white, and BW is the one which we by mistake we have generated the first figure. And default cartoon means the one where you can see alpha helices and beta sheets. And also where, I'm sorry, wait. Okay, you want some dark, you want some mattes, and if you want to play with resolution, so you can uh, change those settings. But remember, first you will write this, the double dash, then render style, and then here we will provide a specific value. Let's say if you want to change the render style in the script is already written below. So you will change, let's say, render style, double dash, render underscore style. And from this render style, which is written in the caps lock, we will write, let's say, default here. We will write default black and white. So the same way, we can tweak this command, we can add or we can remove certain things in this command. So, how you will use this command? If you come here, this command is already written here. If you can see Python pdb.png.py and then pdb file. What is pdb file? pdb file, we have already used it and we have created this entry. Let me show you here where I we have created this file here. Wait. Okay. So in the first step, pdb id, we have created the pdb id and pdb file was also created somewhat here. PDB ID again. Okay, so this is actually the PDB file or the PDB ID is this one which is downloaded here. So the program will use instead of PDB file the same thing. Now, how to change this code? This is important for you to generate good figures. You can see the last line of the figure generation script, Python pdb to png.py and pdb file. So it was the basic thing. Now here you can see in this line usage pdb2.pdb2png.py, and here we will write Python pdb2png.py. Now we will provide anything we want to extract or we want to subtract or we want to delete whatever. So let's say in other example I will teach you, you will better understand. Okay. Now here pdb png render style default. Now, let's say I want from the render style, I will look here. Render style, let's say I want MUTED, muted. So, render style and click here. Okay, one thing you have to take care of, if you generate one figure, let's say we have generated this figure, then before generating another figure, you will need to download that one. The reason is that, okay, it was generated, now we will save it. Okay, so now you can see this is the mutted style. So the reason is you will need to download the previous one because the same file is uh, changed by the new file or the new figure. You can see download, it will be the smarted one. Okay, now, so this is one thing. I tried to change the code for you, and every file should be generated with a different name, but that requires some time, and we are actually using the IPython display, so that's why I skipped the same way that, okay, I will ask you people or request you people just to download the figure before to generate a new one. Okay. Now here we are using a longer script. Let's see pdb python file. Here we want to show the ligand information. So the ligand information, if you see here, let me clear this one, then it will. Okay. If we if we see here, so the ligand ID can be shown, the ligand color can be shown. So and the ligand chain, etc., etc. We are using this one, hit atom color, or then we will provide some color entities, or we also want to change the rotation. So for rotation, I already told you we will use some values. Okay, so I would suggest to read these information. Okay, now you see Python PDB PNG up to this. It's the same code. Last time we have used double dash render style and then default, and on in the second attempt we use BW and mutted. Now here we are going to use hit atom color, that will be red. I mean, I want to see the color of the ligand molecule in red color. If you want to see black, green, whatever, then the ligand ID here is XZ, but I don't know in the protein I use what is the ligand ID. So you just need to open the PDB database 4I5S and just confirm what is the ligand ID of your protein you are dealing with, because at times the protein may have more than one ligand. Then how the program will know that what is the actual ligand in the structure. Okay, I think we do not see any ligand information. Let me check. Okay, I do not see any ligand information, so quickly I will change the structure to some other protein. Let's say uh, once it is installed, you can change quickly. Let's say 4EXS. It has kept a prev, so that's why. And don't run all the cells again, just run the cells which has the information of our structure here. Now it will download the structure here, and the PDB ID is changed. And let me tell you 4EX is a dimeric structure, so we will see two different chains. Okay, so the hit atom color will be red, and now we will see 4EXS. Actually, we want to see the ligand ID from the PDB. Here you can see XZ. So you can refresh. You can see some other PDBs are here. Okay. Now Method. Okay, so the hit atom color will be red. The ligand ID is X8Z. Yes, it's perfectly fine. And the ligand color, once again, this is the hit atom color that is red. This is the ligand color. You can see above that what it explains. And normally these are universal colors. So even if you Google this value, you will see all the values for different colors. And then if you want to rotate protein, let's say I'm using 130, let's say 70, and let's say 15. And render style, I need default. Just click here and en- ensure that you have downloaded your image before generated on the above script because it will replace the existing figure. I have tried, I was thinking that I will finish this video before 15 minutes, but still I'm going with 26 minutes. So I'm sorry because I want to clear each and every point for you because most of my users from their age group I can see in the YouTube section, they are undergraduate students. So for them, I want to clear most of the things. Therefore, I go in extra detail. Okay, here you can click here and it will generate the figure we asked and save the figure. Now you can see these are two different chains, but now you can fix the rotation and this ligand is red because these are zinc and the ligand, these are actually the hit atoms. So the hit atoms, both are actually the hit atoms, but the code will takes the hit atom as the cofactor or the ions or zinc if they are present, and the other the color information 1.1 comma 1 comma 0 above that was for yellow color. So if you change it, you will uh, take some other you will have some other colors and other kind of information. So finally, to drop the final note for you guys, you can generate good quality images, but you will take the information from here, what changes you want to bring. So just play with it and you will generate extremely high quality images very easily and you can use those images. And I also taught you how to manipulate this script. You will just give this name and then you will use the zoom value here. For example, -40, for example, 100, whatever zoom out is in plus and zoomed in is in minus. So use this value protein underscore zoom with this double dash and then some value in this kind of script you will see here in the last line, then you will see the difference. Okay, so I hope you like this tutorial. Thank you so much. Bye bye.